All right, good afternoon. Let's get started. But first I need to go through a few recaps with you. I'm a little bit sick, so my voice is not as loud as normal. If you can't hear me, move to the front. So previously we were talking about the cell cycle, which is basically how the cell spends its life before it divides. Right? When the cell needs to divide for various reasons. It needs to divide for growth and repairment. We need to replace lost or damaged cells. And for some organisms, they need to do cell division for asexual reproduction. Now, there are a couple of key things I need you to pay attention to for this chart because we're going to be emphasizing and elaborating on these things as we progress through the lecture. This kind of pink part right here, that's the interphase. The cell spends 90% of its time in this phase. And majority of the time, it's going to be growing, right? It's going to be growing, gathering resources to prepare for DNA replication. And afterwards, after it copies all the genetic material, it's going to grow some more to prepare for the eventual cell division. Now, what's really important to understand is along the way, you are going to have two major checkpoints here. Okay, we have what's called the G1 checkpoint and G2 checkpoint. And this is a checklist that the cell has to go through to make sure everything is okay before allowing it to progress to the next part. Okay. If it fails at any of these checkpoints, then the cell is basically not allowed to divide any further. Okay. We will look at how these checkpoints plays a role in cell division, but also what happens when they fail. If we don't have these checkpoints in place, or if they're not working the way they're supposed to, then the cell cycle is actually going to spin out of control. We do not want a cell to be progressing through the cell cycle very rapidly, because that means the cell will be dividing and dividing and dividing in a controlled manner, right? So keep that in mind, and we will talk more about it. Now, we're going to be starting today's lesson with mitosis, which is the cell division aspect. Okay, so when it's ready, when it's big enough, when it has copied all of its genetic material, then it's going to progress to the actual cell division. So that's what mitosis is. Okay, it has four separate phases in mitosis: prophase, metaphase, anaphase, and telophase. You can remember the order. I mean, you can memorize. It's only four things. It's not like the end of the world. But if you need a little bit of help, you can remember PMET. Okay, prophase, metaphase, anaphase, and telophase. So we're going to go through each of these phases with you. But first, let's take a look at some pictures, and then we will draw it out to help you understand a little bit better. This is, oh, actually, you know what? Let's quickly talk about it, and then we're gonna go. And this is early prophase. Prophase is the first part of mitosis, right? So P for prophase, and you can think of P as preparation. At this point, the cell is preparing for the division. And I'm gonna dim the light a little bit to show you. I'll turn it back on in a bit, but let it open. Does this weird thing. Anyway, this is an actual picture of the cell, and there are three colors. Obviously, like the cell has no colors, very thin, it's transparent, but they have stained the cell here to highlight certain features. Blue is where you will see the genetic material, and you can write down blue is genetic material. Green is the spindle fibers. So I'll write down for you. Blue is genetic material. Red is basically the cytoplasm. It's where the cell is. And then green is the spindle fibers. If you forgot what the spindle fibers are, they are these cytoskeleton. They're like ropes. The best way to think of them is like ropes. They will grab onto the DNA and they will pull them around in the cell and move them around. So at this point, if you look at the genetic material, can you actually see the individual chromosome? I would say not really, right? It's just like a blurry sea of blue, right? And that makes sense because at this point, the chromosomes are still in the form of chromatin. And you might remember chromatins are the form of DNA that exists when a cell is not dividing. Okay? They are these strands of DNA that are wrapped around those yo-yos, right? Those proteins, and it's packaged into the nucleus.
Can you describe it a little bit more, please?
Describe the chromatin?
Realize picture in my head.
The chromatin?
Yes.
Sure. That's a good idea, probably.
Thank you.
Yeah. So let's take a look again, right? This is the naked DNA that I was telling you about. Right. Just DNA on its own. You don't have to draw this. You can just watch. This is DNA by itself, but it's too much. Two like meters of these things you can't fit into the nucleus. So what happens is you will have these special proteins. Okay. These are called histone proteins. You have this slide already. So again, unless you really want to, you don't have to draw it. And what happens is the DNA will go and wrap it around these proteins. Okay. So that way, like it's more compact, right? Okay. So I think it's like the yo-yo, right? Wrap it around it. And in this form, it is called chromatin. Okay.
Thanks, sir.
Yep. So they are condensed, but they're not like super condensed. So that's why I can't really see it. But they're starting to coil up more. They're starting to condense even more. And when they do it, it will get converted into chromosome. And by later on in the prophase, you can see the individual chromosome, right? You can see the almost like little worms, right? Before you can see it. So it transitioned from the chromatin into the chromosome. That is one of the events that happens in prophase. Okay. Now, of course, we need to have the chromosome coming out of the nucleus, and so we will need to take away the nuclear envelope, and the spindle fibers will now attach onto the chromosomes. So let's draw this out, and probably it will help you understand a little bit better. Switch to today's notes. What is it, March 11th? All right. Now, before that, I need to go through a few background stuff with you to make sure you understand what's going on here. Okay, so let's say we have chromosome 1 here and chromosome 1 here. Okay, we'll say this is chromosome 1 from mom. That's the one we inherited from mother. And you will also have a chromosome 1, which you inherit from father, right? They are both chromosome 1. So 1 and 1, and then you have 2 and 2, 3 and 3, so on and so forth. Number 1 to number 22, they are called autosomes. Remember that word? Autosomes? No. 1 to 22, it's autosomes. Yeah, and 23 is sex chromosomes. So 1 to 22 determines everything about yourself, except for the sex. But for 23, if it's XX, it's going to be female. If it's XY, is going to be male. And so we talked about that. Now, for simplicity, I'm not going to draw out all 23 pairs of chromosomes. Now, you start off with 46 chromosomes, and then you're going to have DNA duplication. And then you copy all the DNA, and we'll see how many chromosomes we have next. It's not what you think. So let's write down here DNA duplication, replication means the same thing. So, humor me. How many chromosomes do you see over here? Yeah, two, right? It's not a trick question. Two chromosomes, right? And then we're gonna have to copy them. And after you copy them, this is what happens. Okay? Instead of just one of the chromosome 1 from mom, now you have two of them that are exactly the same and they will be attached together. And then same thing here. They will be attached at this location. And I'll tell you what the name of that is. Now, how many chromosomes do you see over here? And that's what most people think, right? Okay. But that's not the case. Because the scientists or whatever they decided if they are stuck together, then we will count it as one. Right? So even though there are two pieces to it, because they're stuck together, this is considered as one chromosome. And this is also one chromosome. So even after you duplicate it from the perspective of counting, it is still two chromosomes. Do you understand? You guys know what that chocolate, the Kit Kat, right? It's two bars, right? Yeah, but when they're together, it counts as one. If you break it, now two. Okay. So over here, this is still considered as two. Okay, you should make a note of that. Count as two chromosomes. Okay, so you started with 46. After DNA duplication, you will still have 46. Now, what do you call these copies that are identical to each other? Those are called sister chromatids. They are identical copies. Only the ones that are stuck together are identical, and only those that are stuck together are called sister chromatids. The red one and the blue ones are not the same. Remember, you inherit the red from mom and blue from dad. They're not identical. But these are non-sister chromatids. How about this one and this one? Sister or not? Sister. Sister. If they're stuck together, they are sister. The location at which they are stuck together is called centromere. Location where sister chromatids are connected. You guys have some kind of major assignment tomorrow or something? No? So rarely people don't show up. I don't know what's going on. Anyway, that's that. So you started with 46. After DNA replication is 46. And then you split it. It is still 46 and 46. So it's a little bit weird if you think about it, right? People often think it's 92, but it's not, right? If anything, like if it helps, you can think of it as 46 chromosome and then 92 sister chromatids. Right? Now be careful. Chromatid is TID. And then earlier, we were talking about, what were we talking about earlier? We were talking about chromatin. Okay, so don't confuse chromatin, chromatid, and chromosome. Are we okay with that, or you need me to clarify a little bit? We get okay, excellent. All right, so let's take a look at what happens with the prophase here. Okay, prophase. That's the first part of mitosis, and you can think of P as prepare. Prepare, just getting everything ready, move everything in place. Okay, by the time you get to prophase, you need to understand you finished interphase. Okay, you got through the checkpoints. You have already copied all the genetic materials, and it's ready to divide. Okay, so by the time prophase is finished, you would have these things called the centrioles. We learned that in lecture 3. And centrioles, they send out these spindle fibers. Okay, they're like ropes, spindle fibers. Now I will draw a few for you. Okay, we will have the mother chromosome 1, which is already copied. And then you have mother chromosome 2, which would be a little bit smaller. And then you might have the father one. And then you have the father one. Okay, they're just kind of all over the place. Now, to help you see it a little bit better, this is chromosome 1 from mom, chromosome 1 from mom, sister chromatids. 2 from mom, and then 2 from dad, and 1 from dad. They are all going to be attached to each other at the centromere. And then the spindle fibers is gonna go latch onto them. All right. By the way, just wondering if you were paying attention last class. Okay, the chromosome. You just learned this today. What about here?
Telomere.
Thank you. I haven't even asked the question yet, but that's a telomere. Very good. Okay. Telomere right here. So telomeres are at the ends. They protect the genes that are at the end. Very good. Okay, so here you can see the nuclear envelope it's gone. Chromosomes are out. It's pretty much everything that's on the slide, right? I'm just drawing it out for you to help you understand it a little bit better. So what's after prophase? What's next letter? M. M stands for meta, that's right. Metaphase. M stands for middle, okay? Middle. And so in metaphase, these spindle fibers, these ropes, they are pulling on the chromosomes back and forth. Like a tug of war. You know, the rope, and then people are trying to pull, right? Tug of war back and forth. That causes the chromosomes to move one way and then the other. And then eventually they will all line up in the middle of the cell. So metaphase, middle of the cell. There you go. And then we have the centromere here. So that's pretty self-explanatory. What comes after metaphase? Anaphase, that's right, anaphase. And A stands for apart. The spindle fibers will now pull really, really hard until the sister chromatids separate from each other. Remember, the sister chromatids are identical copies of each other. So you want one copy to go to this cell, this half of the cell, and then the other copy goes to the other half of the cell. So in the end, you get the same thing for both cells. So this is anaphase. You have these little potato tots, the centrioles, spindle fibers. And remember, this is happening to all 46 chromosomes, but it's too messy to draw all of it, which is why we are only drawing some of them. Okay, so just to help you remember what they are, these are sister chromatids. Okay, so let's number them again. This is 1 and 1. 1 from mom, 1 from mom. 2, 2. Similarly. So during anaphase, sister chromatids separate and are pulled to opposite ends of the cell. And then we're done. Telophase. Telophase is basically the opposite of prophase. You can think of telophase, T for tearing. This one's a little bit harder to draw. Let me try. So we no longer need the ropes. We no longer need the spindle fibers. They will disappear. And the chromosomes are done moving. They are at where they're supposed to be already. So we can start to uncoil them and convert them back into chromatin. Right? So like the chromosomes are like much more like strands now, along with the father one. But what's important is each half of the cell gets the exact copy. And then you're going to form the nuclear envelope again. And at the same time, the cell is going to start to divide down the middle. Like taking a big Play-Doh ball and then just pinching it into two. Okay. So chromosome converts back into chromatin. Spindle fibers disappear. Nuclear envelope reappear. And finally, there's a new word here, cytokinesis occurs. Cytokinesis is the splitting of the cytoplasm. Okay, you want to make sure that each new cell, each daughter cell gets roughly the same amount of cytoplasm. You don't want to end up with one cell that's like super big and then the other is super small. And then each of these daughter cells will be allowed to, go through another round. Yeah, go ahead.
I have a question, sorry.
Mhm. Here, bird, born, or bird, bro. Starts to form that. Spell typo. Very good. Yeah,
Sorry.
thank you, thank you. Are we okay? Pretty simple stuff, I guess. Give you a moment to catch up. Okay, so all this stuff is pretty much in the PowerPoint slides. You can probably go through it yourself. There is a mistake here. Zoran just pointed that out. Typo. There we go. Okay, let's take a look at this. Why don't you just talk with people around you and see if you can figure out what phase these are in. A, B, C. Let's see which phase they're in. These are onion cells, by the way. Okay, in the root tip. Okay, what do you think the first one is? Yeah, first one is metaphase. Everything is in the middle, right? Metaphase. How about this one? Anaphase is correct. Anaphase is separating, right? Yeah. This one? Telophase. Yeah. If you have trouble telling telophase and anaphase apart, telophase usually like you can't see the chromosomes anymore, and they're more like round up inside the nucleus. And if I turn off the light, you probably can see that there's a line forming in the middle, right? That's the new cell wall that's going to be separating the two cells. And over here, this one, that is prophase. That's right. Okay. These are still in like interphase. Right. They're not really dividing yet. Like, I can't even see the chromosome. Right. It's still in the form of chromatin. So that's that. All right. Concept check. What's the answer?
Bro. Continue.
Remember to do the fingers so everybody can try it, right? We wrote this down. When does the nuclear envelope dissolve, disappear? It's prophase, right? Isn't it? You gotta take the nuclear envelope away before the chromosomes can come out. Otherwise, everything is trapped. Okay, this one. See if you were paying attention. Is it? Maybe not.
Maybe not.
Uh-huh. Yeah. Yes. Anyone else? If it's metaphase, they are all in the center. They're still stuck together, right? As x's. So that's 8. But then anaphase, you pull them apart. So then it becomes 16, right? It's just like a matter of counting. The answer is C. This is just application questions. We don't have time to take it up. You can think about it, and if you're dying to know the answer, email me. Okay, the next part is very complicated, okay? I'll try my best to explain it. But if you don't get it, just ask questions, okay? So couple of things first. We already talked about the checkpoints, right? The checkpoints are important to make sure the cell has enough resources and that there are no mistakes and errors, and that it's appropriate for the cell to divide, right? But like, who's doing the checkpoints, right? It turns out there are various proteins that are involved in the process of doing these kind of checkpoints. And there are two kinds of proteins in general, okay? Two types of regulators for the cell cycle. Some proteins, we call them growth factors, and they promote mitosis. They encourage cell division. We will call these green light proteins. Okay, you can think of them as like green lights. Green light proteins. Just like driving, if you see a green light, you go, you drive, you don't stop. On the other hand, we have growth inhibitors, and they inhibit mitosis. We call these red light proteins. How do we make proteins? Remember? What contains the instruction to make proteins? Ribosome makes protein, yeah, but what's the recipe? Where's the recipe from? DNA, right? DNA, okay? So our DNA contains the instruction to make these red light proteins or green light proteins, but DNA can have mistakes. DNA can mutate and change. So if the DNA has a mutation, it's possible that these proteins are not functioning the way they are supposed to. Okay, so that's the first thing to understand. Now, what kind of problems are we worried about? If you have a green light, there are two kinds of problems with it. Okay, either it's turned on forever, or it's cannot turn on at all. Okay, which one is more problematic from the perspective of cancer? Which one's gonna likely cause cancer? A green light that's always on, or a green light that doesn't turn on? Let's think about it. Yeah, a green light that's always on, right? If the green light is always on, then the cell will go through the cell cycle rapidly, dividing and dividing and dividing. Not turning on the green light will cause other problems, but probably not cancer. What about the red light? What kind of problems, what kind of malfunction are we worried about in terms of the red light that would cause cancer? Always on or not turning on? Think of what's going to cause an accident, okay? Driving, right? A red light that doesn't turn on, or a red light that's always on? Which one's going to more likely cause an accident? Not turn on or always on? Not turn on, right? Because if it's always on, people just like all stuck there and you're being angry and stuff like that. But if it doesn't turn on, then people don't know to stop and they just keep on going. There's going to be crash, right? And that's what happens here. If the green light is always on, then the cell will keep on going through the cell cycle, dividing. Alternatively, if the red lights are not on, there is nothing stopping it from progressing, and it will also be dividing and dividing and dividing. Does that make sense? Okay, so let's take a look at two kinds of genes. Now, for now, genes are basically, here I'll write down, genes, they are a segment of DNA with instruction to make proteins. All the proteins that we have in the body needs to have a gene that corresponds to it. Gene is, think of it as the recipe to make the protein. If the recipe is wrong, then the protein is going to be wrong as well. So the green light proteins, the recipe for it, the gene to make these green light proteins, they are called proto-oncogenes. Proto-oncogenes. Okay, I'll write down for you. Proto-oncogenes, they make green light proteins. And when they function normally, they will help a cell grow. In a normal cell, it's a delicate balance between turning on the green and turning on the red, at the right time, so the cell will divide when it needs to and stop dividing when it's not supposed to. However, a proto-oncogene, by the way, we all have it, right? We all have these proto-oncogenes, and we all make them. What is going to be problematic is when these proto-oncogenes mutate, when they change, or sometimes you might end up having too many copies of it, then it turns into a bad gene. This bad gene is going to make a green light protein that is going to be malfunctioning, and it might be turned on permanently. And when that happens, the cell will grow out of control, like I told you. This mutated proto-oncogene is now called an oncogene. So oncogene is bad. Okay, proto-oncogene is just the normal. What do people with cancer, what kind of doctor do they see? What do you call them? Oncologists. Oncogene, oncologists, same thing. Okay. This is all right so far? The opposite of a proto-oncogene is a tumor suppressor gene. Tumor suppressor gene, these are genes that makes the red light proteins, right? Make red light proteins. And they generally will slow down cell division. They will do other things like repair DNA. Let's try to fix the error before we copy the DNA. And if it's not fixable, they'll tell the cell to die, apoptosis. So these tumor suppressor genes involved in various processes that will prevent a cell from dividing. That's why they're called tumor suppressor. They prevent the cell from dividing out of control. When tumor suppressor genes don't work properly, the red light they produce will not turn on. Without functioning red light, the cell will not stop and it will divide and divide. Okay. So normal versions are proto-oncogene and tumor suppressor genes. When they mutate, both can lead to uncontrolled cell division. But the kind of mutation is going to be a little bit different. For proto-oncogene, it is called a gain of function mutation. Okay? Another analogy to help you understand is like if you're driving, okay, and we have two copies of the genes, right? One from mom and one from dad, right? And if one of the copy is mutated, it's like having a stuck accelerator. That's enough to speed up the car and cause an accident, right? So gain of function mutation only requires one of the two copies to have an error in order to cause uncontrolled cell division. Right? Once again, we have two copies of the genes. Only one of them needs to be wrong in order for problems to arise. Let me try to explain this with the green light analogy here. Normally, we have both of the copies functioning. So when the green lights are on, do we divide or not divide? Divide, right? Okay. And then when I turn them off, we don't divide. Okay, so that's very easy. All right, never mind the red light for now. Just focus on the green light. You turn them on, you divide. You shut them off, you don't divide. Okay, but what happens is if we have a mutation and it causes one of the two green lights to be permanently on, okay, like this one. Let's say this one is mutated and it's permanently on. So what happens is when you try to turn it off, only one of them turns off, but the other one stays on. And so what happens? The cell will just divide because it sees the green light. Okay, it's the same as driving. If you're driving and then there are two pairs of target lights, if one of them is not working, well, I'm just going to look at the other one. If the other one says green, I'm going to go, right? So only one malfunctioning protein is sufficient to cause and control cell division. All right? That's not the case for the red lights. That's not the case for tumor suppressor genes. For tumor suppressor gene, it's not a gain of function mutation. And you should write this down. It is a loss of function mutation. Loss of function. As opposed to gain of function. And this time, you actually require to have two mutations. Both copies needs to be losing their function before we have a problem. Let's try to go through the exercise again. If the red lights are on, do we divide or not? No. How about now? Do we divide or not? Divide, right? Okay. So what happens is, let's say I have a mutation and one of them is not going to turn on. One of them is not going to turn on, okay? So I turn them off. We divide. Okay, but now we want to stop the division. So I want to turn the lights on. But this one, the one on the right, is not turning on because it has a loss of function mutation. Only one of them is turning on. But will the cell divide or not divide in this case? Again, think of driving. If you're driving through an intersection, one of the lights is busted, and then you see a red light on the other one.
Divides.
Do you go or not go?
Go.
No, you don't go, right? If you see one red light, if there are two sets of traffic lights, and one of them showing a red light, and there's a car over there, do we go through the intersection? No, right? Red light, right? Red means stop, right? Okay. So in this case, even though we only have we have one busted copy, but the other one is working, it will still be functioning to stop the cell from dividing. Do you understand? It's only, and this is very important to understand, okay? It's only when both copies have the same loss of function mutations, then there's no hope, okay? We can't stop the car anymore. We cannot stop the cell, then it keeps on dividing. Okay? So if you don't like the traffic light analogy, think about driving, okay? Let's say your brake's not working for whatever reason. Do you have a backup plan to stop the car? No, really? Do you have your license? Well, other than the foot brake, what other brake you have to stop the car? What other brake do you use? The handbrake, that's right. Okay. I mean, it's not ideal, but like it's it's like a backup thing, right? So if both of those things fail, then I really can't stop the car, right? Do you understand? Okay, so for the green lights, they are actually called growth factors. One gain of function mutation is enough to push the cell over the threshold, so to speak. But for the growth inhibitors, we're going to have two loss of function mutations. What is the implication of that? Okay, so that first 2 points I already mentioned. The last point, I'm just going to read it to you and explain. People born with only one functional copy are more likely to get cancer. Right? Why is that? So let's try to draw it out and see if it makes any sense to you. Okay. So I'm going to have a bunch of cells here. Okay, we have trillions of cells, okay? Cells. These are cells. Now, I need you to think about this carefully, okay? each cell is gonna have 2 copies of these genes, right? Okay, two copies of the red light once again. Right? So let's say the person gets a mutation here, one mutation. Right? Is that gonna cause cancer? No. Yes or no? What do you think? If there is one of the red lights not working in this cell, will we have uncontrolled cell division, guys?
No.
No, right? Because there's still one functioning red light, we're good to go, right? Okay. Now here's the next question that really needs you to test your understanding. Let's say the guy has a mutation here. Okay. Now, will he likely get cancer? Yes or no? Why not? That's right, because in this cell, it still has one more functioning red light, right? It's not both red light in the same cell, right? You know what I mean? Okay? It's like one broken red light here and then another broken red light there. It's like one broken red light, but in different intersections. It's not gonna cause an accident yet, right? Okay. So how are we gonna push this body to potentially have cancer? You need to have the same mutation twice in the same cell. Okay, and that is rare, right? Because we have so many cells, and it's just not very likely to happen. It's not impossible; it can happen. That's why we get cancer, okay? But it's unlikely. Okay, it's like trying to blindfold me, and then I just randomly throw eggs at the wall and hitting the same spot twice. Right? It's just not a very likely event, right? Now, what does the note say? It says people born with only one functional copy are more likely to get cancer. Now, where do we get our DNA from who? From who? Parents, right? Okay. So it's possible we inherit one mutated copies, right? Doesn't mean our parents have to have cancer necessarily, as we will learn why later, but like they might have a mutation that they're not aware of. Okay. Remember, if you only have one mutation, you're not going to show any symptoms, right? And you could potentially pass that on to the next generation. So if somebody inherits one mutated copy at birth, all their cells will only have one functional red light. Okay? And now, if this person has a single additional mutation in any of their cells, that's enough to cause the cancer. You understand?
What is single additional mutation? Can it be like, outside factors? Like
Yeah, that's a great question, right? So we will learn that later. There are many things that could cause mutations, right? For example, drinking alcohol, eating too many processed meats, right? Being exposed to UV light, right? Okay, so if you enjoy, doing a barbecue while having a beer and then, eating some sausage and then your friend is smoking next to you, perfect combination for mutation. Anyways, so this is important to consider because it says, first of all, mutations in cancers could be inherited or acquired, right? But most of the time, sure, environmental factors could increase our risks of getting mutations, but sometimes mutation is just bad luck, okay? You could do everything right, okay? You can eat healthy, you can exercise, and mutation can still occur, right? It's just a random event that happens during cell division sometimes, okay? And I'm oversimplifying things here, right? In the body, in the cell, it's not like we only have a pair of green lights and a pair of red lights. There are multiple growth factors and multiple growth inhibitors, okay? And it generally takes more than one mutation to cause cancer. Okay, but like I'm simplifying it for you to understand that, right? So if you have multiple red lights failing and multiple green lights failing, then yes, it's very likely that it will change into cancerous. Okay. Now here's the important point. When someone has inherited an abnormal copy of the gene, they already start out with one mutation, and then it makes it easier for that person to develop cancer. Which is why cancer that are inherited tends to happen earlier in life compared to cancer of the same type that are not inherited. I'll give you an example. There is not yet. Before the example. Genes is not everything, okay? there's a line in genetics that's called nature versus nurture. Nature is what you are born with, the genetic composition. You can't alter that, okay? But nurture is the environment that you're growing up in, okay? And that's all the environmental factors that we're talking about, right? Okay. So just because you inherited a set of genes that could potentially increase your risk, it doesn't guarantee you will get the condition necessarily. And that's what the concept of penetrance is about, okay? Let's say someone inherited a cancer gene, okay? How likely is it for that person to actually get the cancer? If it's guaranteed, then we say complete penetrance, 100%. If it's only 50-50, then it's incomplete penetrance, right? Okay, so the higher the penetrance, the more likely the gene is going to manifest itself into a disease, okay? And that's based on study of wide population and statistics. People for a while were really into ordering these genetic kits from online, right? You can collect a sample of your saliva or whatever, and then you send them out to these companies and they will sequence your DNA. Do you guys know about these things? Yeah, or like they're 23andMe or something like that. Yeah, and then they would give you a report and then they would say, oh, you have these genes, and some will go as far as predicting when you will die. Okay, I'm always suspicious when people tell me when I'm gonna die. Okay, it's like you have these genes, you're gonna die at 63. I would be very skeptical. Okay, and it's good to be skeptical because those things don't take into consideration of penetrance, right, and environmental factors. All right, so I'm gonna give you an example right here. There's a gene called BRCA1 gene. Everybody has BRCA1 gene, okay? Males have it, females have it, even though the name BRCA1 stands for breast cancer 1 gene, okay? So breast cancer 1 gene or BRCA1 is responsible for repairing DNA damage. And if it cannot be repaired, it will cause the cell to die. Based on this description, you should understand it is a tumor suppressor gene and not a proto-oncogene, right? Okay, because it prevents the cell from dividing. All right. So who has the BRCA1 gene? Everyone. That's right. I just want to see if you were listening. In the general public, statistically speaking, 12% of women will develop breast cancer sometime in their lifetime. Okay, now it's quite high of a number. Okay? However, for women who inherited a harmful copy of BRCA1 gene from the previous generation, if they inherit a broken red light, right, their risk of developing the same type of breast cancer is now as high as 55 to 65%, depending on the ethnicity, for example, and other factors. Okay, but it's substantially higher. We're talking about 5-fold increase in risk, right? Okay, so people understandably might want to know their genetic makeup. This is something you can screen for. You can pay money and they can tell you whether or not you have these mutations. And some of you might know this actress, right? Angelina Jolie. Okay, she famously did double mastectomy, so she removed her breast because she her mother died from ovarian cancer. I think her sister also died of a similar form of cancer, and she did a DNA analysis of her own profile and found that she inherited the BRCA1 gene, the mutated one, okay? And that means she is at high risk, right? Compound that with the fact that two of her close family members die of cancer, that puts her at very risky area of developing cancer, right? And so she kind of did like preemptive prophylactic surgery, right? To remove the breast, and therefore she will not get the cancer, right? Okay, so that actually started like a whole movement in the US, I think, for people advocating for screening. But healthcare experts are saying, this might not be the best way to go about doing it. Number one, it's going to put a strain on the healthcare system, right? And number two, not everybody can actually afford to have these kind of surgeries done on them, right? The US is not the same as us in terms of healthcare, right? Like most of the stuff are covered in Canada because of good health insurance and things like that. But it's not the case in US in most of the cases. She did an interview on this, so if you're interested, look it up.
Just one question. Can cancer reoccur
Absolutely, yes.
even if they did the perfect vasectomy?
Yeah. So, but here's the thing, right? Because the breast cancer happens in the breast tissue, by removing the breast tissue, then the structure where it's most prone to getting it is no longer there, right?
It can also spread, right?
Huh?
It can also spread to like other.
But only if the cancer is already formed. If it hasn't formed yet, then it's not gonna, spread, right? All right, let's keep going. I'm just gonna power through, okay? Like, I mean, it's a small class, so no point taking it. Keep going and then finish it. Now, we understand cancer is uncontrolled cell division. But not all uncontrolled cell division is necessary cancerous. When we have these, red lights and green lights not working, then the cell undergoes rapid division, and that could form a tumor. Tumor, fancy word is neoplasm. Neo means what? Anybody know what neo means? New, that's right. So this is a new growth. That's what it is. Okay, neoplasm. Now, this tumor might or might not be cancerous. Right? People will have to do a biopsy, take a little sample of the tumor if it's accessible, and then to determine whether it is benign. Right? Benign is the good news. Right? If the biopsy come back and say your tumor is benign, it means it's not aggressive. You can usually remove it through surgery, and then nothing will happen. But malignant is the cancerous form. And that's the form that is, Okay. So how do people know like whether it's malignant or benign? It turns out there are a set of characteristics that they use to characterize cancer. Okay. So let's take a closer look. 6 hallmarks of cancer. Number 1. Self-sufficiency in growth signal. They no longer require external stimulation to tell them to divide because they have their own green lights. Okay, like I don't need you to tell me to grow, I'll just grow all the time. Right? I have my own growth signal. Okay. Number 2, insensitivity to anti-growth signal. You tell them to stop, they don't stop, they don't listen, they just keep on going. Like my kids at the back, they tell them, stop fighting, don't hit each other. They can't hear, they wear the headphones. Right? Stop fighting each other. They don't listen, they keep on, fighting. Right? Insensitivity to anti-growth signal. Okay? They grow on their own and they don't stop if you tell them to. Those are the 2 characteristics, right? Okay, please memorize some of these characteristics for your next quiz. Judging from the time, I think I can actually keep the quiz next Tuesday, so I'm not gonna move it yet, okay? We'll see how it goes on Thursday, and then I'll tell you. But for now, it's on Tuesday, okay? Next thing, we have quiz, next Tuesday. Supposedly, yeah, on 6 and 7. Next, they are able to evade apoptosis, right? Apoptosis is programmed cell death. When a cell is accumulating all these mutations, it's supposed to die, but they don't die, right? They become immortal. Now, to supply all these growth is very energy demanding. And so the cancer cell has the ability to create their own blood vessels. That's what angiogenesis is. Create new blood vessels, okay? And these new blood vessels basically leach resources from surrounding tissue, whether it be like nutrients, oxygen, and all these nutrients is going to fuel the rapid growth of the tumor. They also have limitless replicative potential. Remember, because of the telomeres, after you divide so many times, you're supposed to stop critically short telomeres, right? But not for the cancer cells. They have evolved to turn on the telomerase, and that allows the telomeres to reset, and then they can keep on dividing without being senescent, okay, without becoming old. And finally, when they grow so much and they have exhausted the resource at the local tissue, they will just spread. Okay, spreading is called metastasis. All right, so 6 hallmarks of cancer. Yeah, I will remember at least 4 of them, okay, for the quiz. Okay, for the quiz. And that's it. You guys can do these ones later on. That's it for 6, okay? We still have quite a bit of time. Do you guys need a break? No break? No break. Okay, so we're gonna keep going then. We're gonna do number 7. Number 7. Okay, a little bit. Okay, 5 minutes, 5 minutes. Yeah, go. Lecture 7. So lecture 7 is on protein synthesis. Protein synthesis. Now we remember the central dogma of biology, hopefully, which is we started with DNA, and we're going to make a copy of it in terms of RNA, and then we're going to make proteins from it, right? The first part of the process is called transcription, and then it's called translation. So that's what we'll be doing in this lecture. But first, I got to go through a few terms with you to make sure you actually understand when and how to use them. So first of all, there is genome. Genome is all the genetic information of an organism, all the ACPGs in the organism. The genome, you can think of it as like the whole textbook. A gene is a section on the chromosome that contains instruction to make protein. We already learned before the break that you have genes to make green light proteins and red light protein. So the chapter in the book is like the chromosome, right? So you have chapter 1 all the way to chapter 23, right? And within the chapter, you're gonna have different sections. Section 1.1 is gonna tell you about certain topics. The gene is a section on the chromosome that gives you the instruction to make a certain protein. And finally, the location of the gene is called the locus. The chromosome is really long and it has many genes on it. Where is the gene located? That's the locus. And that would be like the page number. The locus, the location at which you find the, let's say, BRCA1 gene, is the same on everybody. Okay? If it's on chromosome 14 for you, it's on chromosome 14 for me. Okay, so these are the different terms that you should be familiar with. Most of them you already know, right? Maybe like genome and locus is new, but gene and chromosomes, we've been talking about it forever. Now, here is another word, okay? I just need to make sure that you actually understand because there's so many terms you get confused, right? So 46 chromosomes, right? Or 23 pairs. These are some important numbers. Number 1 to 22 pair. Thank you. Yeah. Autosomal, we'll say autosomal or autosome chromosomes. And then the 23rd pair 22 pair. What do we call it? Sex chromosome. Okay, now, I already told you chromosome 1 from mom, like our mom and our dad, right? Chromosome 1 from mom, chromosome 1 from dad. These chromosomes are same length, same size, and they contain genetic information in the same order. We call them homologous chromosomes. Okay. Do not confuse homologous chromosomes with what I have over here. These are sister chromatids. They are stuck together. They are exactly the same. Homologous chromosomes are not identical. However, they are same length, same size, and the order of genetic information are the same. Meaning, if on the mother chromosome, you have eye color here, you will find eye color at the same locus on the father chromosome. If you have blood type here, you will have blood type there as well. However, the version of the gene might not be the same because our parents are different, right? I mean, same ethnicity, there are many shared stuff, okay? But like not everything will be identical. Okay? So like if one parent has like type B blood, the other parent has type A blood, then I might inherit different genes for blood type. Right? They might be the same. Right? Both of my parents have brown eyes, and I inherit the brown eye gene. Right? So they could be the same, they could be different. Okay, but the type of genetic information are the same for homologous chromosome. Okay, 1 is homologous with 1, 2 is homologous with 2. Right? And as we go down the list, as we go from 1 to 22, the chromosomes get shorter and shorter and shorter. We arrange it based on size. Okay, I'll show you a picture of that later. Different version of a gene is called an allele. So brown eye gene versus blue eye gene, that's an allele. Okay, brown eye gene versus like the hairline, okay, whether it's straight or wrinkle peak. Right? You guys know hairlines? They could be straight or like wrinkle peak, right? So wrinkle peak, right? That's genetic. That would be an allele, right? Okay, but the hairline, wrinkle peak versus eye color, that's not allele because they're talking about different genes, right? It's only different version of the same gene that's considered as an allele. Do you understand? Okay. So this is the central theme of biology I already told you about. We have transcription and we have an additional step now called post-transcriptional modification. And then we have translation. I'll write it out for you to help you understand. Let's take a closer look at transcription first before I give you the why do we do transcription at all? We mentioned this before, but let's go through it again. DNA is too big, right? It's like 2 meters of this stuff in the nucleus. You're not going to be able to get the whole thing out of the nucleus and try to read the instruction that way, right? In fact, we don't want to take the DNA out of the nucleus unless we absolutely have to, because it might break down in the nucleus. It might degrade. So what do? Instead of taking the whole set of DNA, the entire genome, you're just going to make copies of the gene that you need to form a copy. Now you're going to make this thing called the messenger RNA. Not only does this allow you to control, because you can decide when to copy which gene, right? You have all the recipes to make all the proteins in the cell, but you're not going to make them all the time. You're only going to make the proteins that you need when you need them. Okay, so it allows you to have regulation. You might recall base pairing rule, A to T, G, and C. I mean, you definitely should know that by now. If you don't, then, I don't know what to tell you. The same rule applies to DNA and RNA, but instead of thymine, we have uracil, right? Uracil. So where does transcription take place? Okay. Transcription occurs in the nucleus because that's where you find the DNA. And the enzyme that makes the RNA is called RNA polymerase. Because we're building RNA now. We're not building DNA anymore. It's not DNA polymerase, RNA polymerase. And what it does is it uses one of the strands as a template to create the messenger. And at the end, you are going to get the pre-mRNA, which is not the final product. It requires further processing before going on. There are 3 steps, and I think I'm just going to draw this out for you to help you understand better. I guess you don't need this. Okay, we'll just take this note, and then we'll be done. Okay, and instead of drawing the double helix like for the whole thing, I'm just going to draw a box here. So this here is a gene. It's a segment on the DNA. But how do you know it's the beginning of the gene, right? I mean, all of these DNAs are either A, C, T's, or G's. How do you know where the gene begins and where it ends, right? Well, in the same way, the textbook is all text. It's all alphabets, right? A, B, C, D. How do you know where the chapter begins and where it ends? Well, there are certain cues that would tell you it's the beginning of a new chapter. And maybe it says chapter 1, right? Maybe there's some pictures in it, right? That tells you this is the beginning of a chapter. And so similarly, for a gene, there is going to be a section at the beginning that has special sequences. We call this the promoter. Promoter marks the beginning of a gene. And it is usually AT rich. There are a lot of adenines and thymines in it. Do you remember how many bonds holds A and T's together? 2, that's right. Compared to 3 with C's and G's, right? So if I want to copy the DNA and I want to open up the gene here, if it's rich in A and T, then it makes it easier for you to open up the DNA. Then we're gonna copy the gene if we need to. And how do we know when to stop? It's gonna get to the end here. And this is called the terminator. Terminator sequence. Marks the end of a gene. Now, the process of transcription has three parts to it. Number 1, 2, and 3. 1 is initiation. Number 2 is elongation. And number 3 is termination. What's the name of the enzyme that is going to copy the gene again? Anybody remember? That's right. So for initiation, the RNA polymerase will bind to the promoter. You know, I probably should write down for you. Sorry. Initiation. RNA polymerase binds to promoter and unzip it. Unzips DNA. And then number 2 is the elongation. I'll explain that part in more detail soon. And number 3 is termination. Okay. Sorry. Okay, so I'm going to explain a little bit more about elongation, and then we're going to try a few examples, maybe, and then we'll go. Actually, let me see. Okay, I'll explain once to you guys first, and then maybe I'll explain again at the beginning of next class, okay? So here, the DNA is unzipped right there. Never mind the 5' and the 3' in this case. It's already very complicated. Okay, so let's say this is A, T, C, C, G. And then the other side. It's complementary. This is important part. DNA is double-stranded, right? It's complementary, it has two sides. But only one of them, either the top one or the bottom one, is going to be the oh, sorry, I wrote it wrong. Caught my mistake. That is called the coding strand. Coding strand. Coding strand. Okay. So only one of the two is going to have the actual code that allows you to make the protein. Okay. Don't memorize it as always the top or the bottom. You wouldn't know. I would have to tell you. So on the test question, I'll say the top one is coding or the bottom one is coding. Okay. So we're gonna have the RNA polymerase to come along, and it's gonna make a copy of the coding string. Let's just say, in this example, let's just say the top is the coding. Okay, again, it could have been the bottom. You don't know. I'm gonna tell you on the test. Okay. So the RNA polymerase is gonna come. This is elongation now. Right? It opens up the DNA, and then it's gonna make a copy of the coding strands. So I want you to think about this. When you make a copy of the coding strand, should I build using the coding strand, or should I build using the opposite strand? If I want to make a copy of the coding strand, do I use the coding strand or do I use the opposite strand? Yeah, what am I really asking? Right? Okay, look, this is really hard to explain. I'm so sorry.
It seemed like you were explaining in the last class 5 factorization toward 3 factorization, 3 factorization toward 5.
Yeah, sort of. Okay, but look, listen, right? Let's see, you know, today is warm. If I ask you to copy this sentence, what are you gonna do? You will just copy it, right? Today is warm, no problem, easy. I just copy it word letter by letter, correct? Okay. But as we saw, we cannot, for the DNA, we don't copy letter by letter. It's not possible for our body to see an A and then copy an A, to see a T and then copy T, right? The way we copy DNA is we always build the opposite, right? The complementary nucleotide. So if I want a copy of the coding strand, I cannot use the coding strand because I will end up having something that's the opposite of the coding strand. Which is why, if you want to have a coding strand, you must use the other strand to build off of it. Okay. So this other strand, this being, okay, the strand that is being built off of is called the template strand. Template strand. All right, we're almost done. Almost done.
So the one that is copied.
The one that is built on is called a template strand.
Okay.
And then let's see what's gonna happen, okay? Now, I'm just gonna draw the RNA polymerase here. Okay, let's draw it a little bit better, maybe. Okay, let's say it's going this way. Okay. Now work with me, guys. We're almost done, I promise. When it was moving along, it sees the T. What is it going to build? A. And then when you see a T, what are you gonna build? Try again. U, right? Because we're building RNA, right? Yeah. And the next one? C, G, so on and so forth, right? Okay. And so this here is the RNA, okay? But it's pre-mRNA. It's not done yet, okay? And so at the end, if you finish this process, the RNA that you get is a copy of the coding strand. It's the same. Look, A and A, but T is replaced by U because it's RNA, right? But everything is the same. That's the key thing. You understand? Yeah? if you see your classmate tomorrow, tell that Vincent say good luck, right? So here to learn it. Okay, now, just before you go, I want to show you. It's like making a coin, right? If you're at the mint and you're trying to make a new coin, the mold that you're using is opposite of what it's supposed to look like, right? Otherwise, the coin is going to come out backwards. And that's the idea I need you to understand. If we are copying the coding strand, if we want it to come out like the coding, you must use the flipped strand, the template strand to build it. Okay? So think about that. Next class at the beginning, we're going to be doing these exercises. Okay? These are very important exercises because there will be like a 5-6 mark question on the test, on the quiz that requires you to decode these things. And we will learn how to change it into a protein. Okay? Well, thank you for coming. I'll see you all on Thursday.